Hello everyone, and I want to thank Thermo to invite me to present my research on cyanotoxins and cyanobacteria. I will present you a part of the work that we are using on a large-scale project that is about the monitoring and prevention of cyanobacterial blooms and toxins through genomics. I won't talk about genomics here, it's more about the toxin analysis. And it uses high-resolution mass spectrometry. So cyanobacteria are generally, they appear without showing any real toxicity, but we know that there's more and more intense proliferation of these cyanobacterial blooms, and they are linked to both human and animal intoxication cases. The main cause is the production of cyanotoxins. And the problem is that there's maybe 40% of the cyanobacteria that can produce cyanotoxins. So if we have more bacteria, we have more toxins in aquatic systems. Also, one of the major problems of the cyanobacteria and toxins is that they are practically impossible to precisely predict their presence or production. So this means that for any water treatment systems, it can be really costly. And in United States, the annual cost is about $800,000 million only to establish the ban of drinking water for a period of time. Sometimes it's only to give bottles of water for the inhabitants. So I want to show you what really means a massive bloom. First, we saw in 2011 in the Lake Erie, there was a massive bloom in the south of the lake, and it was the first time that we saw that. And we were thinking, okay, there's something here that is going on. But we thought that it was only one time going on. And after that, we saw in 2014, it was worse, and worse after in 2015. So it really suggests that the climate changes and the agricultural wastes are major factors on the growth of cyanobacteria, and it tends to be worse and worse every year. So we have to do something about it, but it's totally natural. So it's a little bit complicated with the knowledge that we have. So here another problem that we can have here. The water source that we see here is within a water treatment system. And there was a massive bloom inside the water treatment system. And the concentration of microcystins was about one of the major toxins, 119 microgram per liter, considering that the regulations is about 1 microgram per liter. And after the treatment, we saw in the tap water 2.5 microgram per liter. So we couldn't predict. It was a one-time case, and we didn't really knew that it was happening. It was we went there, we saw, oh, there's massive bloom, and it was really problematic. And also really interesting, the US EPA in 2015 suggested a regulation for children protection, and the regulation is 0.3. So with a 2.5, it's a little bit problematic. And now it's on, oh, that's in French, sorry. So overall, we considered cyanotoxins as an emergent threat in aquatic environments, even though we know that it always have been there. But it's more and more present, or we are more and more studying these toxins, and they have a lot of, we are really challenged analytically speaking, because they are really different in terms of physicochemical properties, and also since we don't really know when they are happening and when they are been produced, it's really difficult to really know when we have to analyze them properly. Also, there's a lot of variety and congeners, so it's really difficult, and we have also different variation degradation, different conditions of storage, extraction, analysis is really different for each of the cyanotoxins, distribution and availability of standards, of course, and the analytical costs. So my work was mainly to have a better detection or more specific, and also group the more toxin in one analytical method to be able to be more specific and precise in the detection. And I will present you some of the method that we developed in the lab. The first toxin I will present you is one of the less known, is the BMA, which is a non-proteinogenic amino acid, and possibly, it's still controversial, possibly linked to amyotrophic lateral sclerosis and Parkinson's disease complex called the Guam disease. We know that more than 95% of toxic cyanobacterial are BMA producers, but there's no regulation nor monitoring program for this toxin because for many reasons. There's a lack of knowledge about its toxicity, and also it's really difficult to analyze. So for the previous work on this toxin, the first methods included fluorescence, but tends to give an overestimation on the concentrations. So the main work are on mass spectrometry, both with derivatization to use reverse phase chromatography or helic chromatography. But there's not a lot of analytical methods that can include BMA on a large amount of toxins inside one method, so it's a little bit complicated. Our strategy was to use derivatization using densyl chloride, and also BMA. Another problem we have with this toxin is it has many conformation isomers. So same mass, same protein, same structure. It's really small, really polar, and we wanted to do a derivatization and also include more cyanotoxins that aren't in the same family. So we included Anatoxin-a, salindrospomopsin, and saxitoxin, which are neurotoxins. And we used the Q-exactive for high resolution detection, and also to determine the fragmentation pattern because since we are using a derivatization and we don't really know what the fragments we are going to have in the analysis, we wanted to be really more specific and really study the fragments that we would have with this derivatization. I will go fast on the workflow. We use 100 milliliter of lake water, submitted to electrolysis to leach the toxin. We add the internal standard. We studied the filtration material because it's another challenge with the cyanotoxins. Some toxins can absorb on some material and some are not absorbing on these materials, so we have to study precisely which filter that can be used so we don't lose any material or toxin during our analysis. We acidified the sample and submitted to a solid phase extraction. Dry the sample, reconstituted with the reagent solution for the densyl chloride derivatization, which is really fast, 10 minutes at 60 degrees C. And here are the parameters for the analysis. And what is important here, we couldn't separate the two signals here, the AAG, one of the isomers, and the BMA. So with all the parameters that we were using with the columns, the solvents, we couldn't separate the two compounds. So we used another strategy to be sure that we could be selective. And we used Mass Frontier to generate theoretical mechanisms to hope that we could find specific fragments from each of the compounds, which we did. And it's still theoretical, but with the comparison of the fragmentation pattern, it gives us a good idea that we could we can use these fragments and they are specific to the derivatives. And here we go. So for the mass spec, For the BMA, we used 277. That was in the mass spec of AG, and AG we use a 289. And for the quantitative fragment, and for the confirmation, we also use specific fragments. So we could do the analysis selectively and be sure that we didn't add any false positive. We had some results in lake water that were contaminated with cyanobacterial blooms, and it's the first time in the province of Quebec that we saw the BMA. So it suggests that we have to develop some monitoring programs to include the toxin in other cyanobacterial toxins monitoring programs. Rapidly, I will show you the determination of Anatoxin-A with a really fast detection method. The Anatoxin-A is a neurotoxin that has been associated to animal mortality. And the toxin isn't really stable under natural conditions, but is still, when it's leached from the cyanobacteria, it can be still lethal. One of the main challenges of the Anatoxin-A is, first, it's sensitive to UV light, so we cannot use any spectroscopic analysis. And also, using the LC-MS, the phenylalanine, which is an essential amino acid, is an isobaric interference, both in with the parent ion and the fragments. So we have to be sure that the chromatographic separation is complete, or else we have high interferences. But we use the Q-exactive with high resolution, and the masses were completely distinguished. So it's really helpful for that. We didn't really needed to have a great chromatography. And also we use the LDTD, which is a device that is produced by Phytronics Technologies in the province of Quebec. And this technology gives an instant information since it's really fast. It doesn't use any chromatographic separation. It's only do the thermal desorption. We put the sample in a well. The laser goes back in the well, and with a thermal desorption, it goes right through the mass spec. So it's really fast. So with the resolution of the Q-exactive, we could separate the signals of phenylalanine and Anatoxin-A. And with the LDTD, we could do a really fast analysis. And also, we didn't really need a complex sample preparation. We submitted the samples to a cell lysis, added the internal standard, only did the filtration, changed the pH to acidic conditions, and did the analysis with the LDTD. So really, really easy, really fast. This is the mass spectrum only to show you the signals of Anatoxin-A and phenylalanine, which is, well, the A is, it's really close. Well, yeah. And the second most abundant isotope, so the signals were distinguishable and no false positive were possible during the analysis. Also, the internal calibration that we use only to show you the linearity of the LDTD experiment. And also, really important, our limits of detection are a little bit higher than compared to liquid chromatography, but the regulations for Anatoxin-A is 3.7 microgram per liter, which is much higher than our limits of detection. So we were in the right range. And we did some monitoring on some lake samples. And we compared also the full scan mode with the PRM mode to know which one was best to analyze the anatoxin. And we saw that the full scan mode tends to have more matrix effects and also interferences. So we use the PRM mode, which was better for our analysis. And our last method, which is the more complete one, is the Microcystins analysis. Microcystins are the mostly known cyanotoxins. We know that more than 100 different microcystins are produced and are identified, but I saw just at the beginning of the year we are now at 235 microcystins that have been identified, but only 15 standards are available. So we don't have all the screening of the microcystin when we do the LC-MS analysis. So what we wanted to do is to be sure that we analyze all the microcystins. We wanted to develop an oxidation method to extract the MMPB part, which is here, from the polypeptide cycle of the microcystins. This part is common to all congeners, and with that we can have an idea of the total concentration of microcystins in one sample. Also, the microcystins tends to bioaccumulate and bioaccumulate on fish tissue or animal tissue, which is problematic because a irreversible link is caused within the tissue, and it's really difficult to extract the microcystin from the tissue. So our workflow for this analysis, we used fish tissue, added the internal standard, submitted to digestion to be able to really have a good extraction of the microcystins, submitted to an oxidation, the alumio oxidation, which cuts the ADA part from the microcystins, and we can get the MMPB. We filtrate, submitted to a solid phase extraction, and after that we analyzed it with the LDTD APCI HRMS at this point. So here are, I think it's 25 samples out of 50 that we analyzed for this project. These are fish samples that were taken from contaminated lakes from the government of Quebec, and with 4 standards they only got 1 positive, and we had the half of the samples with positives for microcystins. The 2 suggestions about that is, 1, maybe there were more congeners in the samples, or 2, the conventional extraction are not really effective to have all the microcystins from the sample. So we can suggest that there's more microcystins in the sample that we can get with normal extraction and analysis. We firstly did this experiment with water, and we had the same conclusion. We saw more microcystins using the total microcystin technique compared to an LC-MS analysis with all the individual microcystins. With all these techniques, we were really curious about other samples. We mostly do environmental samples, but we had some dietary supplements in the lab, and we were like, okay, we can analyze them. And there were cyanobacterial dietary supplements, spirulina, and well, this name Flossacque, well, whatever. It's not really the supplements I would take myself. But the surprise was we saw some microcystins mostly, but also Anatoxin-a and BMA in the samples. But the worst part is some of the dietary supplements had up to mostly 700% more microcystins than the tolerable daily intake regulation suggested by the World Health Organization. So when you take a lot of microcystins one time, maybe the contaminated water that you take, okay, you have a stomachache, but this time it's supplements that you will take daily, and these toxins are sometimes considered as tumor promoters, and this suggests that the quality control is not quite there for these supplements, and I wouldn't recommend anyone to take these. So it was interesting, but a little bit wasn't really happy to. Finally, for the project, Our main concern is the automation of analytical techniques. I presented to you more complicated strategy to have a more specific detection, but we want to put all these techniques in Online SPE coupled to UHPLC and HRMS for our larger scale monitoring of cyanotoxins. And we first had this technique for individual microcystins. For now, we have 14 microcystins with low concentration levels for limits of detection. And we do it with the Q-Exactive at full scan mode. And the idea is we don't have all the standards in hand, but we can go back in the data and search for qualitative analysis to know maybe if we do the total microcystins, oh, there's more, we can go back and see if there's any other microcystins or other toxins in the samples. Also, we want to do the same strategy with BMA and include more polar toxins using Analytical chromatography and hopefully with a SPE that could work with it. Gabriel Munoz worked on the total microcystins using the SPE, Online SPE, and the limits of detection were quite low, which is really good for any type of drinking water or really low concentrations of microcystins. And this is only to show you it's not all the microcystins and we don't really see the congeners, but there's a lot of congeners. In red, it's the standards that we have, and the other it's not available. So it's really interesting to use a Q-Exactive in a full scan mode and really have all the data and go back to the data and see if there's any other cyanotoxins in the sample so we can do some identification. Also, we want to include other families like micropeptins to really go to a large-scale cyanotoxin analysis. So it's pretty it, and I want to thank you for your attention. If you have any question, thank you.
You had mentioned that you do an online SPE method for these. What sorbent are you using for the online SPE portion?
It's SC18 from Thermo. It's normal reverse sorbent.
So, and you have no trouble with Anatoxin or you can do BMAA as well?
No, not for this one. That's why we want to work with the HiLic, and we didn't really try it yet because we have to get the online SPE for this. But no, the Anatoxin-a works really well, I gotta say. But when we go to more polar toxins, it doesn't work at all.
Thank you again, Audrey.
Thank you.